Section: Introduction. In this section, we discuss the advancements made in the field of reconstructing visual perception from brain activity using deep learning models and functional magnetic resonance imaging (fMRI) datasets. We have observed a surge in research papers showcasing the accurate reconstruction of visual perception from brain activity, thanks to the availability of deep learning models like clip and stable diffusion, as well as large-scale fMRI datasets such as the natural scenes dataset. fMRI works by indirectly measuring neural activity through changes in blood oxygenation levels. The patterns of brain activity detected by fMRI are then translated into embeddings of pre-trained deep learning models, allowing us to visualize internal mental representations. This visualization of internal mental representations has the potential to revolutionize clinical assessment methods and brain-computer interface applications. Despite the promising results in reconstructing perception from brain activity, the practical application of these approaches has been limited. This limitation is mainly due to the use of single-subject models that are not easily generalizable across different individuals and require extensive fMRI training data. To address these challenges, Eye2 introduces a novel functional alignment procedure. This procedure involves pre-training a shared subject model that can be fine-tuned using limited data from a new subject, enabling generalization to new data from that subject. By implementing this approach, we achieve reconstruction quality comparable to a single subject model trained with significantly more training data. Eye2 also builds upon the previous approach introduced by Eye1, where both methods map fMRI activity patterns to the latent space of pre-trained deep learning models. Additionally, both Eye1 and Eye2 map brain activity to Stable Diffusion's variational autoencoder to obtain reconstructions that excel in low-level image metrics. Eye2 introduces several innovations compared to Eye1. These include pre-training the model on data from multiple subjects, mapping to a richer clip space, merging high and low-level pipelines, and predicting text captions for images during the reconstruction process. These improvements lead to state-of-the-art performance in image retrieval and reconstruction metrics using the full fMRI training data from the natural scenes dataset. In summary, Eye2 involves pre-training and fine-tuning a single model to map brain activity to deep learning model embeddings, enabling accurate reconstruction of scene images from minimal training data. The full pipeline of Eye2 is illustrated in the figure, showcasing the training process on GPU nodes with specific batch sizes and training frameworks. Section Summary. In this section, we introduce Eye2, a method that pre-trains and fine-tunes a single model to map brain activity to pre-trained deep learning model embeddings, enabling the reconstruction of visual perception from brain signals. By leveraging a novel functional alignment procedure, Eye2 overcomes limitations of single-subject models by achieving high-quality reconstructions with minimal training data, even as little as 2.5% of a subject's full dataset, showcasing state-of-the-art performance in image retrieval and reconstruction metrics. Section: Shared Subject Functional Alignment. In this section, we discuss how we align the functional patterns of brain activity across different subjects. Each subject has a unique brain structure and functional organization, so we need to align these variations before processing the data. Unlike anatomical alignment, where each subject's brain is mapped to a standard template, we stay within each subject's original brain space. We align the flattened patterns of fMRI activity to a shared subject space using subject-specific ridge regression. This means that each subject has a personalized linear layer to map the fMRI data to a common 4096-dimensional space. After this initial alignment step, the rest of the model is shared among all subjects without any subject-specific adjustments. We train the entire pipeline end-to-end, with each training batch containing data from all subjects. This approach ensures that the model can handle inputs from different brains effectively. One strength of this method is its simplicity and flexibility. By using a straightforward linear mapping for alignment, we can achieve robust performance even with limited data and high noise levels. Additionally, our approach can handle situations where subjects view unique images during training, unlike traditional methods that require subjects to process the same set of images. Next, we describe the backbone, diffusion prior, and submodules of our model. We first map the brain activity patterns to a shared subject space with a dimensionality of 4096. These latent representations then pass through a backbone neural network with 4 residual blocks before being mapped to a 256 times 1664 dimensional space of image token embeddings. These embeddings are further processed through a diffusion prior and 2 MLP projectors. We use a diffusion prior inspired by previous work to align the fMRI latents with the image space. This prior is trained from scratch and helps map the brain activity patterns to the image embeddings effectively. The retrieval submodule in our model uses a combination of contrastive and MSE losses to learn embeddings that satisfy multiple objectives. By applying different loss functions to different parts of the model, we can effectively train it to perform well on various tasks. Additionally, we incorporate a low-level submodule that maps the fMRI data to a latent space using an MLP projector and a CNNUP sampler. This submodule helps generate reconstructions of the brain activity patterns that are useful for low-level metrics. By including these different components in our model, we can effectively align and process the brain activity data across different subjects. Section Summary. In this section, we introduce a novel shared subject functional alignment method that aligns flattened fMRI activity patterns to a shared subject latent space using subject-specific ridge regression. Each subject has a unique linear layer to map input fMRI voxels to a 4096 dim latent, followed by a shared model pipeline. This approach offers simplicity and flexibility, enabling robust performance in low sample, high-noise scenarios and accommodating unique image viewing experiences, crucial for datasets like the natural scenes dataset. Section: Image Captioning. In this section, we predict image captions from brain activity by converting the predicted embeddings from the diffusion prior's VIT-BIG-14 to CLIP-VIT-L14 space. We then pass these embeddings through a pre-trained generative image to text (GIT) model. Previously, it was shown that using GIT to caption images from brain activity in the natural scenes dataset is effective. To enable this process, we trained a linear model to convert open CLIP-VIT-BIG-14 embeddings to CLIP-VIT-L14 embeddings. This conversion was necessary because there was no existing GIT model that could accept open CLIP-VIT-BIG-14 embeddings as inputs. Predicting image captions from brain activity adds flexibility to decoding approaches and helps improve image reconstructions to match specific semantic content. We then discuss fine-tuning Stable Diffusion XL for unclip. CLIP is a model that maps images and text captions to a shared embedding space. Unclip models work in the opposite direction, going from this shared embedding space back to pixel space. Previous unclip models focused on replicating high-level semantics rather than low-level structures. Our goal was to train a model that can return images closely resembling the reference image in both low-level structure and high-level semantics. To achieve this, we fine-tuned the Stable Diffusion XL (SDXL) model using OpenClip VIT-BIG-14 image embeddings. We found that conditioning only on image embeddings, without any text conditioning, improved the fidelity of the reconstructions. Our evaluation in the appendix shows that the reconstructions closely match the original images. We fine-tuned the SDXL model on a GPU node using an internal dataset and specific optimization steps. The model can output images of different aspect ratios, with the best results observed at a resolution of 768 by 768 pixels. The inference process involves feeding the predicted OpenClip VIT-BIG-14 image latents through the SDXL unclip model to generate pixel images. To enhance image realism, we further refine these reconstructions using base SDXL with text conditioning guidance from predicted image captions. This refinement step improves the quality of the image outputs without significantly affecting image metrics. The final refined reconstructions are obtained by combining outputs from base SDXL with pixel images from the low-level module through weighted averaging. For retrieval inference, only the retrieval module's outputs are needed. Nearest neighbor retrieval is performed by comparing the embeddings from the retrieval module with those of images in the desired image pool using cosine similarity. Section Summary. In this section, we convert predicted VIT-BIG14 embeddings to clip VIT-L14 space and use a pre-trained generative image 2 text GIT model to caption images from brain activity in the natural scenes dataset. By fine-tuning Stable Diffusion XL for unclip, we aim to return images closely resembling the original across both low-level structure and high-level semantics, achieving nearly identical reconstructions from ground truth open clip VIT-BIG14 image embeddings. Section Results. In this section, we utilize the Natural Scenes Dataset, NSD, to examine the brain responses of human participants as they viewed various natural stimuli from COCO. The dataset includes data from 8 subjects who underwent 30 to 40 hours of scanning, with each scanning session involving viewing 750 images for 3 seconds each. Some images were repeated across sessions, with a subset of 1,000 images viewed by all subjects. We followed a standard approach for training and testing, using the shared images as the test set and evaluating model performance based on image metrics from 4 subjects who completed all scanning sessions. Our models were trained in chronological order, starting with data from the first scanning session of 750 image presentations. We assessed the performance of Eye2 using the full NSD dataset and compared it with other fMRI-To-Image models. Eye2 showed state-of-the-art performance across various metrics, indicating improvements in reconstruction quality due to changes in modeling, architecture, and training procedures. Interestingly, unrefined Eye2 reconstructions performed better than refined ones in some high-level metrics, despite appearing visually distorted. This suggests a need to re-evaluate the evaluation metrics used in fMRI-To-Image studies. Human raters preferred the refined reconstructions, confirming their subjective quality. We also evaluated Eye2 performance with only 1 hour of training data, showing improvements in reconstruction quality. Our goal was to demonstrate high-quality reconstructions from a single MRI visit. We observed improved performance with more training data, especially with pre-training. Models trained from scratch with limited data showed instability, indicating the importance of sufficient training data. We also compared predicted image captions with previous work, showing that Eye2 outperformed existing models in generating image captions from brain activity. In summary, our findings highlight the effectiveness of Eye2 in reconstructing images from brain activity and generating accurate image captions. Section Summary. In this section, we utilize the Natural Scenes Dataset, NSD, to analyze brain responses of human subjects viewing naturalistic stimuli. We trained our Eye2 model on the full NSD dataset and achieved state-of-the-art performance in fMRI-To-Image reconstruction, surpassing previous models. Interestingly, our refined reconstructions were preferred by human raters over unrefined ones, highlighting the importance of subjective evaluation metrics in assessing reconstruction quality. Section Image Brain Retrieval. In this section, we explore how image retrieval metrics help us measure the detailed image information in fMRI embeddings. When we have many similar images in the test set, like 14 images of zebras, and our model can correctly match a brain sample to the exact image it represents, it shows that the fMRI embeddings contain detailed image information. Eye2 enhances the retrieval evaluations of Eye1 by achieving almost perfect performance on the retrieval benchmarks from previous studies. Even when trained with just 1 hour of data, Eye2 maintains competitive retrieval performance. To compute the retrieval metrics, we aim to identify the correct brain activity sample that corresponds to a given image from a pool of brain samples. We convert the scene image into an open clip or clip image embedding and calculate the cosine similarity between this image embedding and its fMRI latent representation, as well as 299 other randomly selected fMRI latents in the test set. Success is determined if the cosine similarity is highest between the ground truth image embedding and its fMRI embedding, top 1 retrieval performance. We repeat this process 30 times to consider the variability in random sampling. For brain correlation, we assess how well a reconstruction predicts the original brain activity that triggered it. We use encoding models to analyze the relationship between images and brain activity, offering a unique measure of reconstruction quality. This alignment is independent of the stimulus image, allowing us to evaluate reconstruction quality even when the true image is unknown, making it applicable to various domains including mental imagery. Brain correlation metrics may better align with human judgments as they are rooted in human brain activity. We then discuss the improvements of Eye2 over Eye1 through ablations. Eye2 shows better performance without pretraining on other subjects, indicating enhancements in model architecture and training procedures. Changes such as using a linear layer for voxel mapping and mapping to open clip big image latents improve performance across metrics. Training Eye2 with multiple objectives yields mutually beneficial outcomes. In related work, aligning subjects' brains to a shared space is common in fMRI analyses to enhance statistical power and assess scientific findings' generality. Various approaches exist for functional alignment, with some relying on shared stimuli and responses for alignment mapping. Eye2 stands out by demonstrating that a single neural network model can be pre-trained across subjects with unique stimuli and effectively fine-tuned to a new subject with minimal data.